Okay, well, good evening, everybody. I'm really pleased to welcome you all to this evening's Institute of Neurology Inaugural lecture. As you all know, the inaugural lectures are an important event where the Queen Square Institute community come together to celebrate the major academic achievement of becoming a professor at UCL. We've been doing them online in recent times, obviously because of COVID, but hopefully it won't be too long before we can actually do the events face-to-face again. So this evening, I'm delighted to introduce Professor Andrea Malaspina, who has been appointed Professor of Neurology and Clinical Academic Lead of the Queen Square Motor Neuron Disease Centre. I would like to just highlight that the development of this appointment was a very collaborative effort between partners in the hospital, particularly Chris Turner, partners in the BRC, the UCLH BRC, through Nick Wood, along with myself and the Institute of Neurology. So I'm really grateful to Chris and Nick for making this possible. So as is traditional, I'll say a few words about Andrea. So he trained in medicine in Pavia in Italy, graduating in 1990. He then did his specialist neurological training in Italy and got his CST in clinical neurology in 2000. He then did his PhD in molecular neurobiology at Imperial College London in 2001. He became a consultant the same year, 2001, at Barts and the London NHS Trust, and became clinical lead of neurological services at Basildon Hospital, running a very busy clinical neurology service and developing his interest in motor neuron disease. In 2015 until currently, he's the MND Research Director for the North Clinical Research Network. And he became professor at Barts, the Blizzard Institute, Queen Mary University London, in 2019. He has a major track record in research neurodegenerative disease, and in particular ALS, motor neuron disease. He secured over £5 billion in independent research funding over recent years. He's led in the creation of some very important biobanks and biorepositories for neurodegeneration, which have been a really important platform for research in ALS and other diseases, and has an international reputation for his work in ALS biomarkers in particular. He's also had a major role internationally and also in UK-wide consortia for developing clinical trials and clinical trial readiness in motor neuron disease, which is really important. And he has a really extensive and excellent publication record going back many years now. So we're really delighted that he's now heading up our sort of clinical academic program in motor neuron disease across the Institute and the hospital, working closely with our excellent colleagues, both basic science and clinical in motor neuron disease. It's a very important development for Queen Square and the Institute. And so I'm really delighted now to welcome Andrea from his lounge at home to give us his inaugural lecture. So thank you, and over to you, Andrea.
Thank you very much. I guess you can see the slides and you can hear me. Yeah. Fantastic. Thank you very much. I have to say I'm honored to give this inaugural lecture, and also a bit puzzled by the situation of not being able to be face-to-face with most of you. When you do this from home, you have to sort out different things, including, the delivery and the cat flap just around the corner. So I hope we manage to neutralize all potential confounders. So what I'm suppose, and I will try and do, is to give you a bit of insight of the historical development of my interest in clinical and academic neurology. And I will put some other bits in it, including memories and perceptions of my experience as a traveler, if you want, to the UK. So this is where I essentially come from an educational point of view. So University of Pavia is a piece of historical beauty and architecture. And what I essentially retain as a memory from that period of my life, which hasn't really stopped as I go back to Italy very frequently, is this geographical or rather winter landscape element of fog, which is something like being in a different exoplanet where you have a different atmosphere. It is very unlikely if you think or imagine what Italy is about in terms of landscape, but this is what is more in my heart. It is also a place of historical and architectural beauty, and particularly the area around Pavia. And one of the touching things is the number of Roman ruins or fortifications that are around Pavia. This is a bridge near a little town called Bobbio that crosses the Fiume Trebbia. And you can see here the map of Hannibal army traveling through Europe. And essentially, this is where the Romans had to erect this fortification to try and stop him. And they're still there. Obviously, this bridge is being rebuilt. We all know how it went for the Romans. Having spent 20 and more years in a country like the UK with excellence in terms of teaching and research, it gives me real pride to talk about Pavia, spend a few words about the excellence and innovation that is still present in this university. There are a number of individuals, among whom also Nobel laureates, perhaps a bit dated, but you can recognize here Alessandro Volta. If you're interested in buying a new electric car, you have to pay tribute to this researcher and scientist. We all know Camillo Golgi. Camillo Golgi pupil, Casimiro Mondino, was a neurologist who founded the institute where I train, Fondazione Mondino, approximately around the time of the Italian unification in 1861. Antonio Scarpa, we have a few parts of our anatomy that go after his name, including the vestibular ganglion. And personally, I think that Carlo Cipolla is the most representative. He's an economist. He worked at the London School of Economics and at Berkeley University in California. And what this guy contributed to in economic science, I think is this booklet, which I would suggest everybody to read, about the fact that most catastrophic events in human history are essentially due to human stupidity. And you can plot yourself or eventually whoever you want in this kind of a scheme in which people can be valued for their effectiveness according to whether they spend time to their own good or somebody else's good, or they work more towards improving what is around them. So these are those individuals have been instrumental in my growth as a clinical neurologist, who is still in me the love for the neurological examination. They're not all from Pavia, they're from different universities in Italy, but the ethos of this place has been essentially the ability to go across discipline and look at problems from different angles. Particularly Mauro Ceroni, to whom I owe my development as in the first steps of clinical neurologist, but also as a person and perhaps as a free thinker, as I think of myself. Just to give you an example, Mauro Ceroni and Professor Savoldi, who unfortunately died a few years ago, have embarked in I think in a 10 years writing spree of a book about consciousness seen from the eyes of a neurologist, a philosophist, and from a neuroscience perspective. And I'm not sure it is translated in English, but you might try and find it. It was there and then that I started looking at neuromuscular disorder, and particularly motor neuron disease, as one attractive field of investigations. We all know about the disease, and we know that there are features that can be picked up clinically that are very peculiar and specific to this disorder. So still at that time, in a situation in which we knew very little about genetics, we were expecting certain elements of the clinical development, particularly the strong heterogeneity, the site of onset, the progression of the disease that was very different from individual to individual, as strong prognostic elements. And above all, what we could see at that time was that age was a risk factor, as this was a risk factor for many neurodegenerative disorders, but particularly of a more severe development of the disease. Following the guide of Carlo Rubbia, another Nobel laureate from Italy, from Pavia, who actually contributed to the discovery of the boson particle and was also famous for his quotes, I then moved on to where the best work can be done. And I ended up in the UK with a fellowship that allowed me to work collaboratively between the University of Pavia and University of London. I had the opportunity of working within Jackie de Belleroche group and to access one of those most vibrant environment for neuromuscular and ALS research at that time. Unfortunately, Jackie de Belleroche died a couple of years ago after a short illness. She will always be missed. What happened was, for me, a complete immersion in a new world of neurologists. And you can see some of them reported here. Professor Kennard and Dr. Gilloff were instrumental in my immersion in this new clinical world. Just to give you an example of how things tend to be ramified across Europe, Henrietta Hill, now Bowden-Jones, was another trainee under the same clinical trainee programs in Pavia under Mauro Cerroni. And she was actually slightly junior than me in terms of medical training. And she came to the UK in the same year, and we met by chance in Charing Cross Hospital. And look what has happened to her. She took the path of psychiatry training, become an honorary professor at UCL. She's been awarded the Psychiatrist of the Year prize by the Royal College of Psychiatrists. And she got an OBE for services to addiction treatment and to research. Etta runs the only, I think, gambling clinic in the UK. And she has done an amazing career path. And I really have to congratulate her and hope to see her soon. One aspect about being a neurologist and traveling around Europe is cultural neurology. We are all accustomed to what, for example, cultural psychiatry is, which is concerned with problems related to psychopathology, behavior, and their cultural element. Whereas cultural neurology is about neurologists, not about patients. So in your neurological examination, when, for example, you are eliciting your cutaneous plantar response, you could try and elicit a Babinski sign, and Babinski is a universally recognized sign. but the way you are performing this varies according to different techniques, which go under the name of different neurologists in Europe. Some of them German, some of them French. The same as for the Hoffman signs. I, for example, prefer doing it in the Wartenberg way, which is a bit more forceful, but essentially better informative or more informative. People are still questioning the place of birth of Babinski. Some individuals think he was effectively born in Peru. But each country has its own neurological framework and cultural patrimonium that you arrive and you discover when you had your training somewhere else. Talking about science, what I did in Jackie's lab was twofold. Essentially, I made my decision, and that was something possible within Jackie's group, to whether look at a genetic component of the disease, or whether try and deal with a more dynamic systems like molecules in the blood, or eventually gene expression. I decided to do both. The interesting thing is that I came across with a number of samples taken from individuals who had ALS, coming from the same region of Central and South Italy. It was clearly a very high number of affected individuals in a small area, and this was clearly against the relative rarity of these disorders. And so what we did was essentially to genotype them for what was possible at that time, and we discovered a genetic mutation that was the common factor across these individuals. But to emphasize the characteristic of this disease, among these individuals who share the same genetic defect, there was a very high variability in clinical progression and date of onset, age of onset, and the common denominator across these individuals was essentially the fact that the older individuals tended to have a much faster progression of the disease. So once again, the aspect of age as an element not to be underestimated in this pathology. The second aspect of my research at Imperial College was essentially more exploratory. Jackie had a collection of brain and spinal cord tissue from individuals with ALS from healthy control. So what we did at that time, we essentially started the first process of gene expression analysis, which was essentially based on spotting cDNA on filters and hybridizing using P32. Actually, it's a bit of a barbaric approach if you think of the laser-based detection that you have now with microchips or microarrays. But nevertheless, it gave rise to a number of interesting hits in terms of differential expression. And you can see them listed in this table. The second aspect of this discovery experiment was about pulling out sequences that contain either trinucleotide or larger expanded repeats, unstable regions that were essentially transcribed and expressed in the spinal cord. This was along the line of trying to understand whether ALS had an unstable repeat type of component in the disease. And we pulled out a number of genes. Some of them were not known. They were blastable, but not yet named. Some of them were actually well defined, like the FAS TLS gene, which later on has been found to carry a mutation in individual with ALS. So little by little, based on the available knowledge and also the result of our experiments, we started sort of putting together this puzzle, at least having a glance of what was the main pathological activity or landscape of the motor cell demise in ALS. And this included elements like vacuolar or endoplasmic reticulum changes, mitochondrial damage, certainly alter cholesterol metabolism, and impair axonal transport, and above all, oxidative stress, which was directly related to mutation of the SOD1 gene, which was the first gene found in LS to bear mutation in individuals, mostly with a familial component. The other thing that we were understanding increasingly was the fact that motor cells had a particular structure that made them possibly more vulnerable to external stress, like high energy requirement, and possibly high concentration of structural protein like neurofilaments that could have interfered with axonal transport. The thing that was essentially very difficult to understand at that time, and I think even now, is what was the role of other elements like, for example, the corticospinal tract, sensory afferent, and eventually the neuromuscular junction in the development of motor cell disorders. So the question I had in mind from a very early stage of my career was what was the role of age in the progression of neurodegeneration, in consideration of the fact that many of the scientific element or milestone identified at that point of scientific progress in M&D research were hinting at features that were modifiable by age. At that time, I moved across London. So I finished my PhD in molecular neurobiology under Jackie de Belleroche, and I got into a completely new environment, having made the choice of pursuing a clinical career above all. You can see the structure and the architecture of East London, Barts and the London, the new building just built at the Blizzard. And again, Roman ruins, this time within hotels, so encapsulated within building. And what we did at that time was to start thinking of how the best standard of care could be delivered to patients with MND. So we founded the Barts Health Care and Research Center, which was actually opened by Princess Anne, and it was a very nice ceremony. So the idea behind a care center is about multidisciplinary care, which is known to prolong life for patients with MND. You can see here on the left a number of individuals with whom I've worked very closely, and you will realize that the type of work of a care center was closely intersected with neurologists, like-minded neurologists, MND neurologists across the board. So my collaboration with UCL neurologists was already there and then. Particularly Alex Radunovic, with whom I've shared the difficulties of setting up a service which served a very large area and had to essentially combine the contribute to different specialists and different type of therapists. This was not easy, and it was made even more difficult by the geography of North East London and Essex. See, the M25 in that part of the world is impenetrable. So what happens is that essentially patients that come from Essex, and most of our neuromuscular and MND patients are from Essex, had a significant delay in their transport, most of them coming by ambulances if they had already significant degree of disability. The only way to get to the Royal London safely is like this. Unfortunately, this is something that is limited to certain type of pathologies, but this was a major issue for us, to the point that sometimes we had a clinic of 3-hour duration, and all patients were coming all together towards the end of the clinic. You know, the little signs that physicians had in the corridor in front of their door, stating the delay, the expected delay of the patient. So that caused a lot of gastric acidity, which luckily didn't get into a proper ulcer. But we managed to make this work by putting services into district general hospitals, thanks to the fact that we had job share between District General Hospital and the Royal London Hospital. Another point of importance is the fact that that area is an area of extreme ethnic heterogeneity and, unfortunately, of mixed features of social deprivation. So it has been for me an immense honor to be able to work as a physician, as a doctor in such a diverse environment. If you're traveling from Essex to North East London, is really a travel through different continents. The Royal London Hospital is essentially sitting in the middle of this heterogeneity. You can see here colleagues who have essentially supported me and provided the ground for my growth as a neurologist, and also given me a lot of insight about cultural neurology and the actual way of districating yourself from different situations. The Blizzard Institute, where I was practicing as a honorary researcher until I got a formal academic position, was also an important part of this extreme variability within the clinical and research spectrum. The Blizzard Institute houses groups from different avenues of medicine, and people have done really great things. And one of the things that you can essentially learn from being in a place like this is that you can get a lot of clues from individuals who do a different medicine from yours, in the sense of addressing different type of pathologies. Gavin, John Priestley, Adina Michael Titus, Klaus Schmierer, and Mike Walsh, who's an eminence in M&D, from whom I've learned a significant amount of clinical and scientific wisdom in looking at this type of medicine. You can see here also the M&D team, Brian Dickey, who's leading the science part of the association. And in this specific instance, we essentially gave them shelter during one of their walks through most important institution in London dealing with M&D research, and this was essentially for fundraising. Gavin Giovannoni, I think it's probably not needed for me to introduce him. He's been essentially an important guide in my scientific development. Is an extremely acute clinician, and his comments, the way he elaborates around patients and science is always a matter of learning for people around him. He's obviously interested in the neuroinflammatory aspect in MS, but I guess his development is probably towards a broader concept of what makes the brain healthy and preventative neurology. And I think this is something very relevant, important, particularly in these times. The William Harvey Institute, you can see here some individuals with whom I've worked, from whom I've learned, Sean Henson, Mauro Peretti, Tim Warner, who was the previous director of the Blizzard Institute, Hova Nissin, and Jess Daly. The William Harvey Institute is actually a powerhouse in inflammation, except that they have both an organ and systemic type of approach to inflammation. And but what I get from what I see in their scientific endeavor is that perhaps inflammation is a unifying factor across different organs. This is a big question, and is very relevant also for neurodegeneration. The William Harvey Institute and the Blizzard, probably needless to say, have been instrumental in population genetics, in the development of projects like, for example, the East London Gene and Health, which looks at unraveling rare genetic variants in the context of morbidities within ethnic groups. So in other words, trying to understand the protective or noxious effect of certain genetic modification. And forgive me for doing this, but by that time I got married. Essentially, Cecile is the reason for any professional advance and development, but particularly for my resilience. We have 3 children. They're a bit older now, so they one by one they're flying the nest. And I have to say that I share with them this kind of way of not being able to be serious when somebody takes a pictures of me. So it's something I can't avoid. So we haven't got many presentable pictures. I have to apologize to Cecile about that. One of the things about marrying Cecile is that she has a French mother and a German father. So in my 20 years and more with her, I really became European in the sense that I have now an extended family all over Europe. And I'm like the man this quote is referring to, a quote from Winston Churchill, a man that now finds himself at home everywhere in Europe. Going back to research, what I took the opportunity of developing while I was at the Blizzard, thanks to the advice and guidance of John Priestley and Adina Michael Titus, is to use their model of spinal cord compression to elaborate around what makes neurodegeneration, loss of nerve cells and axons, an irreversible process. So the molecular point of no return. Essentially what we did, we took a rat model of ALS carrying a SOD1 gene mutation, a rather unstable model, I must say. So the miniaturized version mice are much better. And we applied a compression of the spinal cord in a situation in which they were behaviorally asymptomatic. Obviously, biologically, carrying so many copies of a SOD1 gene mutation makes you histologically, biologically already altered. But what we wanted to see is whether the susceptibility to develop a disease, which is encapsulated in this animal model, gives you clues about the biology of the disease. And for example, if we look at it behaviorally, you can see that the animal that got the hit and was wild type, so with no mutation, and a steep recovery in a scale in which higher number are better performance. Whereas the animal model, which was essentially normal before the contusion, didn't recover. And after a while, the disease would start. So the hit was essentially provoking the development of the disease. These animals were essentially vulnerable to the stressor. You might ask yourself why we did this. The reason was to be able to look into the behavior of cells and understand what type of inflammation is behind this worst outcome. And letting aside innate response, one of the things that really captured our attention was the fact that motor cells had a completely different way of behaving in terms of letting off of structural protein like neurofilament heavy chain. So for example, at 24 hours after the hit, motor cells from animal models had a much higher enrichment of neurofilament heavy chain. Both then would lose this enrichment of neurofilaments. And this was for us a starting point to look at structural proteins as a potential biomarkers of neurodegeneration. Certainly it was for me. People were already looking into that, Gavin included. But essentially, the second question was, could we essentially measure these neurofilaments in biological fluid as a readout of disease progression, not only after an injury, but for example, in a progressive neurodegenerative disorder like ALS. And this was a bit of a starting point, turning point, sorry, for me, in the sense that we got funded by the M&D Association and by another charity to work on the animal model. And this essentially prompted my collaboration with the excellent world of basic science in motor disorder in the UCL. Particularly, you see here Linda Greensmith and Lizzie Fisher. And one of my postdoc, Dr. Qinghua Lu, who was initially PhD student, then postdoctoral research assistant, now is a senior academic neurologist in Taiwan. So what we did, we first of all developed a method for measuring neurofilament heavy chain. This was what Qinghua did. And we essentially tested their expression in biological fluid, and we could clearly see that the levels were increasing steeply with the disease progression. And in parallel, you could see the loss of motor cells in SOD1 mutated mice. We could also look at it from a perspective of potential surrogate of treatment response. So we gave this animal arimoclomol, which is a substance that makes chaperone proteins, heat shock proteins, increase. And it's currently being tested in a clinical trial in ALS, in human ALS. And you can see that giving arimoclomol was reducing the area under the curve, which was defined by the release of neurofilaments in blood. So if you want, one of the first proof of the potential as readout of treatment response of a biomarker like neurofilaments. And that was for us an interesting observation, but also something that encouraged us to look into this dynamic field of biomarkers, dynamic in the sense that you're dealing with molecules and with the difficulties related to measurement. ALS was essentially the privileged side to look into biomarkers because of the spread of the disease from the cortical structure to the neuromuscular junction, the number of potential genetic mutations that had been found by that time, and the dense knowledge in terms of pathobiology of the disease. Obviously, the idea was to get a biomarker that was fit for purpose, for early diagnosis, to measure disease progression and response to treatment. The first thing you have to do, I guess, when you're interested in the natural history from a biomarker's perspective of a condition like ALS, is to set up your test bed, which is essentially a longitudinal cohort of biological samples collected from patients that come to your clinic. This is not an easy task. Remember the difficulties in dealing with patients within a multidisciplinary approach, patients that become easily tired and afraid. But we did it nevertheless. And so I think under my lead and of individuals who have become prominent in the ALS field, like Pamela Shaw in Sheffield, Martin Turner in Oxford, and Mike Benatar in the US, also Mary Louise Rogers in Australia, and Pietro Fratta, who's now become officially a colleague. This experiment had been done in different direction and ensure that the ALS research community has a material to work on. The idea was to try and understand what differentiate in terms of biomarker this cohort of patients who have a completely different progression of the disease. Those patients, just to put it simple, that come to your clinic and you see dissipating and getting to end stage in a few months, compared to those individuals who have a similar phenotypic expression of the disease, but can sometimes survive more than a decade. What makes the difference between the two? Another important aspect which we have developed with the monumental work of Mike Benatar in Miami and CREATE Consortium and the PREFAST Consortium of which we are part is the pre-symptomatic aspect. So the fact of recruiting individuals who are known genetic mutation carrier because they are within families of affected individuals who are set to be seen on a regular basis and sampled until they develop the disease. So this was for us the source of both biomarker works, exploratory work, and also of more basic activity like stem cell research. Methodologically, you have to measure neurofilaments. We've done a lot of work, and I have to thank all these people listed here. There are a lot of pictures in these slides, but the balance is between talking and thanking and showing. So I'm trying to do the best I can. So Jens Kuhle, who was a PhD student under Gavin and under me as a second supervisor, this was a bit misleading because Jens is actually a very senior neurologist, is now leading the neuroimmunology lab in Basel. Developed a way of or method to measure neurofilaments in blood when most of the brain protein are easily measurable in CSF, which is the reservoir of any product of neurodestruction. Jens found the right antibody, put it on to an ECL or electrochemiluminescence platform, and the first measurement were done quite easily. Then probably with the contribute of Herrin Zetterberg, who doesn't need any introduction, a CMOA or single molecular array took over with a much higher level of sensitivity. What we have done, and you can see here still Qinghua Lu and Vittorio Lombardi, who's a very active and capable postdoctoral research assistant in my group, is to put these assays into screening of neurofilaments in our cohorts. And we actually produce a lot of data, which I'm going to mention in a second. Another important thing is the extension of this immunodetection approach to a more standard proteomic. And this is the work done with Ian Pike and Proteomic Science, who has actually produced a novel idea of proteomics, which is based on putting tissue and fluid in the same platform and using tissue as an internal calibrant. And with TMT labeling, you're managing to bring the signal of very low abundance protein to a level in which you can actually identify all the fluid proteins that are also expressed in your tissue, a concept which is very important for biomarkers. Axel Petzold, I have to thank Axel. I think is one of the mind behind the science of neurofilaments. So we not only looked at the utility of neurofilament measurement for clinical stratification in ALS, but we also look at the behavior of neurofilaments in biological fluids. So Axel has modeled the expression of neurofilament isoform, particularly the high abundance of light chain versus heavy chain, as the result of a sort of shift of the translational machinery of the cells, which, to save energy, produces a leaner, cheaper version of neurofilaments. That's why you have this increase in light chain and not so much of heavy chain. It's obviously speculation, but it's based on a coarse modeling that is essentially very interesting. With Ian Pike, we've looked at the expression of neurofilaments in blood from the point of view of their propensity to aggregate. So in other words, we managed to develop a system to pull out aggregates from blood and to test whether these aggregates contain neurofilament in significant concentration. And this concept is fundamental if you want to understand the matrix effect that the fluids have, and also any sort of confounder in the expression of neurofilaments. It might also be, and we published a paper recently about this, that circulating protein aggregates may have a role in being conduit of brain proteins, and may have also a function in essentially spreading the disease or making it a more diffuse process. Just to give you insight about what neurofilaments are in across the border, the paper published by Gavin Giovannoni about neurofilament being the CRP of the neurologist is something I essentially I agree with. So in different neurological conditions, particularly in ALS, you can see the versatility and the utility of this biomarker. First of all, and probably uniquely across different biomarkers tested in neurodegeneration, there is a very high level of correlation, so that you can see this linear regression between expression in CSF and expression in blood, which makes it obviously very usable as biomarker. Then, if you want to look at whether it is a good biomarker of disease progression, you probably would say it is in the context of how the disease goes from the pre-symptomatic to the symptomatic stage. This is the work we've done with Mike Benata with different type of neurofilaments. And you can see this rise of neurofilaments in the prodromal stage of the disease. We had only a few patients at that time. Now we have more patients, and we can look into that in more detail. In terms of diagnostic accuracy, with a measurement in plasma, you separate your patients from your control reasonably well. And quite interestingly, if you are interested in different motor cell disorders, you will also notice, according to a couple of publications we put out in the last 2 years, that individuals with SBMA, which is a motor cell disorder by definition, although it's not properly a form of MND, have very little neurofilaments in blood, whereas traditional markers of muscle injury like creatine phosphokinase seems to have a better reading in SBMA. So is this a muscle disorder, or is this more towards neuromuscular junction? This is an initial observation that is worth looking at. In terms of prognosis, neurofilament light chain are essentially better performing than other functional measure in separating your patients who have a higher risk of mortality from those who can survive longer, based on dichotomization and above median and below median, like you see here. One thing we have processed over time is also the importance of neurofilament expression with age. And this is another paper from Jens Kuhle, which I think is fundamental in understanding the importance of age in neurodegenerative processes. So neurofilaments are essentially the readout of axonal loss, like little bricks released by the disgregation of the architecture of the neuroaxonal unit. And you can see here that neurofilaments then increase with age, particularly over the age of 60, where they become more variable, more dispersed, and much higher. And this, as shown in this paper, where neurochemical measurement was associated with imaging and neuropsychological assessment, you can see that this is perhaps the outcome of some subclinical biological processes that become more comorbid in that age group. So what we have done is we have looked at this also from a different angle, and we've recently been funded with James Cole, who works for the Dementia Research Center and for the imaging department at UCL, to combine a neurofilament approach with a structural imaging approach in trying to understand whether age can be harnessed as a potential biomarker of disease progression in ALS. So James has basically used the predicted age difference, which is essentially the biological age that you can infer from using structural imaging like MRI scan from the chronological age as a numerical factor that can be used to test whether age has any effect in terms of differences between biological and chronological age in the progression of the disease. So we are at the moment trying to combine neurochemical measurement and imaging in a kind of multimodal approach to develop a novel biomarker of disease. What we have done in terms of understanding disease progression is to apply TMT calibrated proteomics to both animal model and the human model, dissected in terms of speed of progression. There are G93A carrier animals that can develop a different speed of progressions, like our patients that have a completely different rate of development of the disease. We applied the study of biological fluid using white cells from the same blood sample as trigger. So in other words, we have looked at biomarker in the fluid and protein expressed in cells. And one of the things we have identified in this, I would say, interesting approach is that there is a strong activation of molecular pathways linked to glucose uptake, metabolism, inflammation, but above all, cell senescence, both virally mediated and replicative senescence. And this is something we can see very neatly, both in human and in animal model. So this work has been done by Irene Zubiri, who's now an academic in Spain, and Ella Leone, who's actually still working on proteomics under Ian Pike. And Rocco Auditori, who was also my PhD student, has been very instrumental in developing this concept, which has essentially led us to a second obvious experiment, which is taking the same PBMC and doing immunophenotyping with markers that are able to differentiate cells that are normally functioning from cells that have a loss of effector function or are senescent. And in this regard, the work and the guide and the advice from the William Harvey Institute scientists like Sean Hansen, but also from Timothy Tree, who's an immunologist from King's College, has been instrumental. So we use a multi-dimensional 32 markers, time of flight, mass site off, to test different molecular features of cell senescence in our PBMCs. You can see them summarized nicely here. That's what we more or less see: an increase in memory T cells. There is a change in the proportion of frequencies of CD4 to CD8, but above all, we see an increase in CD57 expressing cells, which is really a marker of cell senescence, which is related to exhaustion of the cells and put in relationship also to viral infection most of the time. And so we were very surprised to find this, given the fact that we have very little knowledge of a potential virally mediated pathogenesis in ALS, except if you consider the strong ground of experimental evidence showing that human endogenous retroviruses, in particular HERV-K, are activated in motor cells from patients with ALS. This is a paper from Avindranath, published in Science Translational Medicine. We have looked also at the other component of lymphocyte element in peripheral blood, which are B cells, which is something normally neglected in ALS research. Somebody did an experiment on animal model in which you could knock out most of the B cell element in the animal model of ALS without essentially modifying the disease. So they thought this was it. There's no B cell element in ALS. But what we see is essentially the hallmark of senescence in B cells. So we have an increase in late memory B cells, an increase in IgG, IgA, but quite interestingly, you can use, again, high level of late memory B cells to predict a reduced survival in these patients with a simple univariate Kaplan-Meier type of analysis. This work has been done by Özlem Yildiz, who's a neurologist from Germany, who's now actually completing her training with 1 year psychiatrist in the UK. She found herself, like me many years ago, the full immersion on the UK NHS quite challenging. And just to conclude about the sciences, so we have essentially extended our interest in the cell senescence mechanism into looking at other factors that are important in the regulation of inflammation or chronic inflammation, like specialized pro-resolvins mediators, which are essentially lipids that are now at the center of interest because they are the tap with which you can stop inflammation. And we found interesting signals in both T and B cells, and particularly at the level of their receptors, which we think could be another mechanism for stemming inflammation in ALS. The humoral response to brain proteins, I have essentially hinted earlier on at ALS as a condition in which there has been a significant body of experimental evidence showing a decrease in T regulatory cells. So the actual mechanism that keeps autoimmunity in place. So we started measuring, for example, the antibody response against neurofilaments under the assumption that this could be a low-intensity autoimmunity phenomenon that may have relevance in the pathogenesis of the disease. Others are looking, for example, at antibodies against TDP-43 with the same mindset. And we actually found that they can be used also for disease stratification. They are good biomarkers, except that obviously their antibodies are stable. They can be measured in small quantity of plasma. This is something we are pursuing. Also, with regard to dipeptide repeats, which are the product of expansion of the C9orf genes, which is among the most commonly mutated genes in mostly familiar form of ALS. And this is probably, I'm verging towards the end. There is no reason to hold such an overview on my research progression without sort of touching base on clinical trials, which has always been something at the back of our mind. Actually, something Gavin has always prompted me to think of. We were always a bit taken aside by the disease, by the fact that it's such irreversibly fatal condition that is discourages you sometimes from thinking laterally, or perhaps this is not the case, but essentially there has been luckily a rebirth of interest in clinical trials in ALS, particularly with the first in human clinical trials using gene modification or ASO techniques. I have to mention people I'm working with, Professor Julian Gold, who's an infectivologist, who actually driving with all his forces this attempt to use antiretroviral to cure ALS, to treat ALS. And Amaral Chalabi, Nigel Lee, Chris McDermott, and Chris Shaw, we all know them. They are important people in the field of MND. These are individuals I work routinely on a regular basis, and from whom I've learned significant amount of wisdom, if I can say so, in research. And Vincenzo Libri doesn't need an introduction. I think he has set up a unit which is exemplary in the way you put experimental neurology and in the context of a clinical trial setting. He has a wit of understanding what is important in a clinical trial. Somebody I'm more and more close to in terms of learning and developing the future of clinical trial in ALS. Just to finish, I think the most important aspect of my contribute wants to be me thanking patients. It is something that is difficult to put into the right words. ALS touches affecting individuals, but I think it does affect many people around these individuals. And it is something that you experience every day in clinic. It's something that you never get used to. Those who spend their life to raise funds and awareness about the disease, you've already seen this picture. This is Brian Dickey and his team. These guys are getting the money through fundraising and trying to put it through channels that ensure the best peer review type of research. They have to deal every day with disgruntled researchers who think that they might have the best idea. But also my friend and colleague Sandy, who leads the South Essex MND research, MND branch. Essentially, Sandy spent her time fundraising in order to then provide what is needed to this patient, which is the cough assist machine, the ventilator. And what she does also, she comes to my clinic and she sits with patients in the corridor when they have to wait, unfortunately, to see me. And she does it sometimes in silence. Thank you, Sandy. And I think I would leave it there. So this is a graphical representation of the Queen Square MND Center. It's been done with Cecile, with my wife. We're trying to put together a few different pieces, but it gives you the idea what I see as present and future for MND under my lead. And I think I will try my best to continue this, essentially this path of research and clinical excellence that I've been witnessing and contributing to. Thank you very much.
Thank you, Andrea. That was a spectacular lecture, and we're delighted to have you as our clinical academic lead at Queen Square. We're not in person, so we can't clap in person, but we have got technology to assist us, and so we will demonstrate our applause for you. Great. So that was a great lecture. So I'm very pleased now to hand over to Gavin Giovannoni to give the vote of thanks. Gavin, thanks.
Well, thanks, Mike. Well, on behalf of UCL and all the online attendees, Andrea, I'd like to extend a very hearty vote of thanks to you for enlightening us about Pavia and your journey from Italy to UCL. I just want to say a few words about Andrea. You know, Andrea, when I was a teenager, one of my favorite books, one of my favorite writers was John le Carré. And his first book was The Spy Who Came In From the Cold. You may have read it, about a British Cold War spy who takes on a dangerous assignment before retiring. Well, I like to think of you as one of those academics who's come in from the cold. Not only have you managed to adapt to the cultural complexities of moving from a foreign place like Italy to London. I mean, I did it from South Africa to London, and at least I was from an English-speaking country. I think it was quite remarkable. And then to work as an NHS consultant before becoming an academic. And how you've managed to, and now you've managed to rise to a pinnacle, which is amazing. I really do admire you for your resilience and perseverance. Well done. I actually met Andrea when I became chair of neurology at Barts in London in 2006. And that time, Andrea was a full-time NHS consultant. He was working between Basildon and the Royal London, and his specialist interests were MND and peripheral nerve disease. And from the very first week of when I arrived here, Andrea stood out as being pretty exceptional, actually. Although he's quite quiet and contemplative and doesn't say much in clinical meetings, he is one of those clinicians who has the ability in grand rounds to cut to the chase by highlighting the key fact in a difficult case, or asking the one question on which the clinical diagnosis or management pivots. So he's an incredibly sharp and skilled clinician, and I have little doubt that your clinical skills, Andrea, will not go unnoticed at the National. And I think Queen Square are very lucky to have you as such an astute clinician. I also want to point out that despite having a full-time and a very busy district general hospital neurology commitment, you managed to carve out an academic career for yourself, which is quite remarkable. So for those of you who are academics, you will appreciate the road from academia to NHS consultanthood is a very wide and well-traveled one. Going back from academia to NHS work is a safe haven often when your constitutions are not strong enough to handle the academic environment. However, the opposite road, going from being a full-time NHS consultant to a full-time academic, and then to become a professor of neurology at arguably one of the, if not the most prestigious academic neuroscience centers in the world, is a very tortuous path. And very few people dare to trod that path. Andrea, I don't know if you appreciate how difficult it is to go from a full-time academia, a full-time NHS, to becoming an academic at Queen Square. So it's amazing that you got there. Just to say, before I took over, just to give you an example, my predecessor, John Priestley, managed to get Andrea a few part-time PAs, program activities. And I think if I remember correctly, you had one or two. So you had a half a day or 1 day a week to do research. During that time, you basically transformed MND services in Essex and East London, and you set up this research network, and you started your biobank on a half a day or a day a week, which is quite incredible. And I think people don't appreciate how hard it is to set up and how thankless it is to set up research infrastructure. And I think you are now reaping the benefits of all your hard work. You are no doubt one of the leading, if not the leading, MND biomarker researcher in the world. And I think you deserve to have the plaudits. I want to just let UCL know that a few years ago, Andrea was being headhunted simultaneously by a medical school in Switzerland and also a medical school in the south of England. Which prompted me, as the center lead at the time, to get an urgent business case passed to pull Andrea out of NHS consultanthood and to make him a full-time academic, a full-time senior lecturer then, and he became a professor about 2 years ago within the Blizzard Institute. So we almost lost him to Switzerland. We almost lost him to the south of England, but we lost him to UCL. But it's around the corner, and I'm hoping that we're going to continue to collaborate, Andrea. I think that if you got more time for research, I think your ambitious body of research is important. And I think you do really interesting stuff from the basic science to the clinical. And I don't want you to lose that. Please keep that in mind. I think you're one of those people who learns from being a clinician. And you personally taught me about rapid and slowly progressing MND, which I always doubted existed, but I am convinced that they are two different entities. And I also think you have a systems biological approach to MND. You have incredibly deep understanding of the basic biology of the disease. And I don't think many clinical academics, in my experience, understand basic biology as well as you do. And I want to point out that your collaboration and work with Denise Shear and Johan Aram on the RNA protein aggregates and also your work on neurofilament aggregates are really pushing the boundaries and asking fundamental biological questions. And I know there needs to be more work done in their area, but I suspect some of your future work will explain some of the fundamental aspects of neurodegenerative diseases. And I hope you continue to collaborate with Denise on this. The other thing I'd like to point out is that you're quite a technophile. I mean, you're one of the few people in our center to adopt new technologies as they come out, be it the Aptimas, the mesoscale discovery platform, or the Samoa platform. And I'd like to thank you for helping us get these platforms into our institute for research. But I think your most enduring and powerful trait is your ability to rise above the political fray and to connect people. And to connect groups. I think that is a really, really important attribute. You know, successful academics, particularly at a national and international level, have that ability to connect people. And I'm sure in 20 years' time, you will be acknowledged as possibly the quiet one, or the one who came in from the cold, but somebody who managed to develop and build an international M&D translational research network. And I'm almost certain it'll lead to new insights, new ways of diagnosing and monitoring the disease. And I think more importantly, new treatments and improved outcomes for people living with MND. So a personal message, as someone who has watched you grow from being an NHS consultant to a professor of neurology and now MND clinical academic lead at UCL, I'm very proud to be associated with you and honored to be part of your extraordinary journey. And I clearly would like to wish you well in the many miles you still have to go in your research career. So I wish I could raise a real glass of champagne, but I can't. So I'd like to congratulate you on your new position and to thank you for such an engaging lecture and an engaging journey. Good luck, Andrea.
Thank you very
Thanks
much.
very much, Gavin, for that. I'm sure we all echo the sentiments you expressed, and we definitely would hope to enable more collaborations with your teams and the colleagues that you mentioned over the next years. So it just remains for me to thank Gavin and to thank Andrea for an outstanding inaugural lecture, and we're looking forward to working closely with you and collaborators over these coming years between the hospital and the institute. So unfortunately, we can't take you for a drink, but you can go to your kitchens and get a drink from the fridge. But that's really the end of proceedings for this evening. So I want to just thank you again and thank everybody. There was well over 100 people attending, which is great. And that's a great sign of interest in what you're doing. But that's all for tonight. So thank you, everybody. Hope you all have a great evening and goodbye. That's the end.
Thank you very much.
Gavin, bye.
Take care. Cheers.
Thank you, Gavin.
Pleasure.